project is great because essentially I have a unique question which is being asked of the project, and we have our lab techniques which we need to use, and these are the same lab techniques that are used in any lab across the world if you're doing this kind of thing, and to a fairly large extent it's on me to work out how to put these techniques together to answer the question I've got, and that's brilliant. That's exactly the kind of thing I expect to be doing if I follow a career in research. I went to a supervisor in an area that I thought would be interested in neuroscience, in this case molecular neuroscience, as it's a molecular degree, and I gave him an idea. But of course he came back to me and said, okay, that's a great idea, may have been pandering to me a bit, but we can't practically do all of that. How does this sound? Could you work something with this topic? And so he gave me a new topic on some mice with a particular mutation in a gene, and it was very interesting to me. So I was quite happy to go with it, and so came up with the title and the proposal for the project, and he was quite happy to go with it. So we set away over the summer working on my third-year project, and we're well into it now. And it's just great to be doing it as part of an actual project, which.
And you're contributing to something really, aren't you?
Well, yeah, it's picking up from where one of his old PhD students left off, and so it's a completely novel research project, which it has literature around it, but it's not just a repetition of a paper which says we found this and we're looking to find the same thing. It's completely, you could write new stuff on it. So
really, if you kind of get the results you want to, and you're actually really quite good with the techniques you're using, it could be sort of publishable in the end,
Quite a big if, but
theoretically,
yeah. And what about you, Jade? How are you finding your project?
At the moment, I did a bit of research on the cell line I was going to be using, which was CDC12. And originally, I was actually meant to be trying to find out the sort of protocols to use in the new machine we have in our lab, which is the new electron transpirator. And it turns out I did already find a protocol for that. So I'm actually moving up in the ladder. My project's evolving a bit more. I'm now using these H2KB cells, which are actually immortal at 33 degrees only, and this is for using a mouse model. So these are my mice cells for Duchenne muscular dystrophy, so they actually have the gene knocked out. At the moment I literally just started culturing them as of yesterday, so I kind of went into a level 2 room, and I had to wear a special coat, special gloves, wash everything down with 70% ethanol. And
like important.
I did. I felt like a big girl, big science girl. I've actually just been asked to join in with meetings with the postdocs and the professor for my project right now, which I kind of feel privileged for, because they're actually kind of acknowledging me to actually want to hear everything that's up to date going on with their project, because my project ultimately is contributing to a postdocs right now, and I get to even use their enzymes, I get to use their cells, I'm going from there. So they're kind of hoping that, at least I'm hoping I don't mess up and then mess them up, but I feel fairly confident that everything's going to go quite well.